Hello friends, and welcome to the lecture series on edible vaccine. In the first couple of lectures, I have discussed about the etymology of the term vaccine and how vaccines came into existence. Then I discussed the definition of a vaccine and what are its different types depending upon its use or method of preparation. I also discussed about the ideal character of a vaccine, what are the limitations of the conventional vaccines, and then I discussed about the edible vaccine, that what are edible vaccines and how these vaccines were introduced by Dr. Charles J. Ernst, followed by discussion on their methods of preparation. And in that, the main focus was on the production of transgenic plants using Agrobacterium tumefaciens. That's how the TI plasmid of the bacteria is manipulated to introduce the desired gene into the plant genome. Then I also highlighted how transgenic plants can be produced by recombinant or the chimeric viruses. After that, I discussed about the direct method of gene transformation, which includes the gene gun or the ballistic gun method, and how it leads to the nuclear or the chloroplast transformations, and why chloroplast transformations are considered better than the nuclear ones. I also discussed about the electroporation method for gene transfer and how electric current is used to create reversible pores in the plant membranes through which the desired gene or the DNA fragment is introduced in the plant. And then we also discussed about certain advantages and limitations of certain methods. So friends, in continuation with the previous lectures, today I'll discuss about the mechanism of edible vaccines. So as I told you in my previous lecture that edible vaccines stimulate mucosal immunity unlike the conventional vaccines. So in order to understand the mechanism by which mucosal immunity is stimulated, we first have to understand different types of immune system in our body and how mucosal immunity works. So coming on to the types of immunity in our body. Now we can categorize the immunity on the basis how the immunity is acquired by the body. And on the basis how it is acquired, immunity can be active immunity or a passive immunity. Now active immunity is that immunity which is obtained after vaccine or the microbial antigen or infection is inflicted in the body or the vaccine is injected in the body. Now, since the vaccine is an antigen, or if our body gets infected by a particular microbe or a pathogen, our body start producing antibodies against it. And these antibodies, they not only neutralize the infection, but by virtue of the memory of our immune system will create memory against that particular infection. So if our body is again exposed to such kind of pathogen or microbe in future, our body will have antibodies, will start immediately producing antibodies against that particular infection, and our body will not get infection from that particular microbe or the pathogen. On the other hand, passive immunity is something which is obtained when serum of a person who has already been immunized, and his serum, which already contains antibodies, is injected to another person who is suffering from injection, and those antibodies that are present in the serum will neutralize the infection and protect the person. Say for example, we get a vaccine against flu infection, and this vaccine will help in generating antibodies against the flu infection, and we will get protected against flu infection. And on the same time, our immune system will also develop memory against that flu infection or the flu antigen, and it will develop antibodies. And so this is nothing but the active immunity. So it will have specificity as well as memory. Now consider one person who has already been injected with the flu vaccine and he has antibodies against flu infection. Now we take the serum of that person, and this serum will obviously having antibodies against flu, and this serum is injected into another person who is suffering from infection or who is liable to suffer from flu infection. Now these antibodies will immediately neutralize the flu pathogens, but they will not create memory of this infection. So injecting serum rich in antibodies into into an infected body or into a body who is liable to get infection is a passive type of immunity. So immunity developed by this method is a passive immunity. Now we can also classify immunity or immune response on the basis of its specificity. And on that basis we have innate immunity, which is considered as non-specific. It will just act on the microorganism, and irrespective of the type of the microorganism, it will show its action. Whereas the second type is the adaptive immunity, which is also known as specific immunity or the antibody mediated immunity. So as the name indicate, antibody mediated. So antibody mediated means we get this immunity when vaccines are injected or antibodies are produced. And since antibodies are specific for specific microorganisms, such kind of immune response is also known as specific immune response or specific immunity. And since this immunity is adapted with the help of antibodies, it is also known as adaptive immunity. Now I will just go discuss briefly about these two types of immunities. Now as you can see in this picture, the innate immune response includes an epithelial barrier defense. It also includes certain antimicrobial peptides, certain chemical barriers, and a complement system, which is nothing but the inflammatory system. Now the innate system, it also involves certain aspects of cellular immunity, and these components or the cellular immunity component includes granulocytes such as neutrophils, basophils or mast cells. It also includes monocytes, macrophages, dendritic cells and natural killer cells. So all these cells constitute to the cellular aspect of the innate immune system. Now the cells of innate immune response or the system are rapidly initiated and they are considered to be the first line of cellular defense against invading microorganisms. Now the innate immune cells, they provide protective immunity against infectious microorganisms until the adaptive immune response can be initiated. Or in other words, we can say that these innate immune responses or immune cells, they also help in initiating an adaptive immune response which is more specific. Now how it works basically, innate system or innate immune response or innate immunity works. Now the innate immune system, it recognizes the molecular structures that are characteristics for microbes or the pathogenic microbes, but they are not present on the mammalian cells. Now the microbial substances that stimulate this innate immune response are called as pathogen-associated molecular patterns or PAMPs. Now different class of microbes, whether it is a virus, a gram-positive bacteria, a gram-negative bacteria or a fungi, all these different types of microbes, they have different PAMPs or pathogen-associated molecular patterns. Now these PAMPs can include nucleic acids that are unique to microbes, such as double-stranded RNA, which is found in replicating viruses, or specific DNA sequence, which is found in bacteria, or they can be certain proteins that are specific for microbes. Now these PAMPs are recognized or they are identified by certain receptors which are present on the epithelial barrier. And these receptors which recognize these patterns or pathogen-associated molecular patterns are known as pattern recognition receptors because they recognize the pathogenic patterns or the molecular patterns. And the most common examples of these pathogenic recognition receptors are toll-like receptors, which are known as TLRs, and these TLRs are the most extensively studied receptors. And there is another class, we call them as nucleotide binding oligomerization domain, that is node-like receptors or NLRs. Now these TLRs are expressed on numerous cell types, including macrophages or dendrite cells or epithelial cells or the lymphocytes, which are T or B lymphocytes, and they also help in initiating the adaptive immunity. Now, whenever the TLR recognize a particular pathogen or a PAMP, it in case of innate immune response, it will immediately initiate a classical pathway, which is nothing but an inflammatory pathway, and we know that inflammatory pathways are defense mechanism of the body. So a classical pathway is initiated by the TLR receptors, which activates the transcription factors like NF-kappa B, and transcription of cytokines such as interleukin 1, interleukin 6, interleukin 12, and TNF-alpha. Thereby, all these are nothing but these are the pro-inflammatory substances that help in initiating the inflammatory response, or we can say a defense mechanism. Now, apart from recognizing the PAMPs, these TLRs or NLRs, they also recognize DAMPs, that is damage-associated molecular patterns. Now, how these DAMPs are produced? Now, whenever a pathogen attacks a particular cell or the epithelial barrier, some of the cells get damaged by the stress or by their action or by the enzymes that are produced by those bacteria or pathogens. Now, such damaged cells, they constitute cellular debris, and all these things, they constitute DAMP. And these DAMPs are recognized by the PRRs, pattern recognition receptors, which are nothing but TLRs or NLRs. And once these DAMPs are recognized by these receptors, they initiate a protective mechanism, that is stimulation of macrophages or inflammatory responses, and also stimulate secretion of cytokines, which also activates the adaptive immune response. And this is how basically they exhibit the protective action against various pathogens. Now, another important property of TLRs is they can discriminate or differentiate between the body's own molecules or own cells or the useful bacteria and the foreign molecules or the pathogenic microbes. And once they are able to differentiate between the useful or the friendly bacteria or the pathogenic bacteria, they keep the immune response in check. Because if they lose this ability to differentiate between the useful and the pathogenic bacteria, then our immune response or the inflammatory cascade will continuously keep on working, and it will start damaging its own cells, which we normally observe in autoimmune disorders. So TLRs also help in maintaining the cellular homeostasis. Now we move on to the adaptive or the specific immunity or the antibody mediated immunity. Now, this type of immunity is stimulated whenever the host is exposed to a specific microorganism, and this microorganism stimulates the production of antibodies that work specifically against the microorganism, that particular microorganism. Unlike innate immunity, this adaptive immune system or immune response produces memory for that infection. As we discussed, that innate immune response does not produce any kind of memory against that infection, whereas adaptive or specific immunity, it produces memory against that a particular kind of infection or a pathogen, so that whenever it attacks again, the body will automatically produce specific antibodies and will show protective action. Now, as you can see in the picture, we can categorize adaptive immunity into two types: humoral adaptive immunity and the cellular adaptive immunity. Now, humoral immunity is mediated by molecules in the blood and the mucosal secretions, and these molecules are nothing but these are antibodies. So in simple words, we can say humoral immunity is mediated with the help of antibodies, and these antibodies are produced by the cells called as B lymphocytes or B cells. Or in other words, we can say that humoral immunity is controlled or is generated by B cells which produce antibodies. Now, these antibodies, they recognize the microbial antigens and neutralize the efficacy of those microbes and target the microbes for their elimination by various effector mechanisms. In principle, if we talk about humoral immunity defense mechanism against, it shows defense mechanism against extracellular microbes and their toxin, because these secreted antibodies can bind to these microbes and toxin and assist in their elimination. Further, these antibodies can also cross the epithelial barrier into the lumen, and there also they can cause the neutralization effect of those microbes by binding them, by causing phagocytosis of those microbes, or by taking them into the cell and exposing them to the natural killer cells. Now, antibodies themselves are specialized and may activate different effector mechanisms. For example, different types of antibodies promotes ingestion of the microbes by the host cells, which I talked about, that is phagocytosis, or they bind to and trigger the release of inflammatory mediators, wherein they promote the inflammatory cascade, and they are actively transported into the lumen, which I have already discussed, that they actively transported into the lumen of the mucosal organ and through the placenta also, to provide defense against the ingested and the inhaled microbes, against the infections even in the newborn babies or babies that are present in the fetus. Now we move on to the cellular immunity or the cell-mediated immunity. Now this type of immunity is mediated by T lymphocytes, which are also known as T cells. So humoral immunity is mediated by B cells or B lymphocytes, and cellular immunity is mediated by T lymphocytes. Now intracellular microbes such as viruses or some bacteria, they sometimes survive the first line of defense and they proliferate inside the phagocytes and other host cells, where they are inaccessible to the circulating antibodies. Now the defense against such kind of infections or such kind of pathogens is the major function of the cell-mediated immunity, which promotes the destruction of the microbe residing in the phagocytes or the killing of the infected cell to eliminate the reservoir of the infection, because these infected cells, they act as reservoirs for the pathogens or the infection causing microbes. Now if we talk about, or if we look into various features of adaptive immunity, as you can see, there are certain features like specificity, we have already talked about, diversity, because this adaptive immunity has B lymphocyte and T lymphocytes, so it has a diversity in the action. Then adaptive immunity also has an ability for remembering the type of infection, therefore memory is another important feature. Clonal expansion, because these lymphocytes, once the body senses the pathogen has entered, these lymphocyte adaptive immune system gets activated. These B and T lymphocytes, they differentiate and proliferate, thereby causing their expansion in population. Then specialization, because all these generate response that are optimal for the defense against different types of microbes. So this is kind of specialization. Then once the immune response neutralizes the pathogens or the infection causing microbes, there is a decrease in the immune response, which is identified as contraction and homeostasis, because one has to suppress the immune in order to protect the normal cells. And another last important feature is the non-reactivity to the own cells or the self cells which are present in the body. So this can be further be explained by this picture. As you can see, there are two pathogens, X and Y. Y is represented by the green color and X is represented by the purple color. So whenever these two pathogens enter into the body, the adaptive immune response will immediately produce antibodies by the B cells against the X pathogen and Y pathogen separately. This indicates the specificity. And then these antibodies or the killer cells will neutralize this Y pathogen and the immune response decreases. This leads to the homeostasis. And finally, memory cells are produced. Now, if there is repetition of the infection, say by the X antigen, you can see there is another peak in the picture which represents the secondary infection. And by virtue of the memory cells, the body will immediately start producing antibodies against the X pathogen, and this time the antibodies that will be produced will be much larger in number, and immediately they will neutralize the pathogen. Now, if we talk about the action of the lymphocytes, it can be categorized into different aspects, and these are antigen recognition, wherein these lymphocytes, they recognize the pathogenic antigen, then lymphocyte activation, wherein they differentiate into different types of lymphocytes, like B cells will differentiate into B memory cells and mature B cells or the plasma cells, which will produce antibodies, and T cells will differentiate into T helper cells, T killer cells, and T regulatory cells, which regulate the excess of immune response. And not only differentiate, these cells also proliferate. That is, they increase, they keep on dividing, they keep on colonizing, and they will increase their population. Next step is the antigen elimination. Now, in this, both B cells and T cells work hand in hand, and therefore humoral immunity as well as cell-mediated immunity, both these types of immunity help in eliminating the antigen or neutralizing the antigen or the pathogen. And once the antigen is eliminated, then the immune response decreases, which is the contraction or the homeostasis phase. And a memory is being created for this kind of infection. And this memory is again, memory cells are produced by both type of lymphocytes, that is B lymphocyte and the T lymphocytes. Now we move on to the mucosal immunity. Now most of the pathogens, they do not get entry directly into the blood, and by not getting the direct entry into the blood to induce the central immune response, but they gain the entry through the mucosal sites, and therefore mucosal immune tissues have developed certain specific or I will say sophisticated defense mechanism to combat these pathogens, and therefore the mucosal immune system is much more larger than the central immune system. But there are a few basic questions about the mucosal immune system or the mucosal immune response. That where is this mucosal immune system located? What are its components? How do it not act against the useful bacteria? How the mucosal immune system works, and how the lymphocytes reach the target site, because lymphocytes, they are largely produced in the lymph nodes, and how these lymphocytes through the systemic circulation reach to the desired or the required site of action. So we are going to discuss each and every aspect. Now the first one is that where is this mucosal immune system located? So any tissue that secretes mucus will have a mucosal immune system. Thus, we can find this kind of system, mucosal immune system, in the respiratory tract, in the gastrointestinal tract, or in the urogenital tract, because all these sites, they are rich in microorganisms. They have a high population of microorganisms. Now, one thing is common in all these locations, that now apart from the high population of the microorganism or the microbes, all these sites, they are exposed to the external microbes or the microbes that are present in the environment, because all these systems, all these tracts have openings. In respiratory tract, it has an opening of nasal cavity. GIT has two openings, that is the buccal cavity and the anal region, and similarly urogenital tract, which has opening at the in the reproductive organs. So all these systems, all these tracts, they are exposed towards the external or the microorganisms that are present in the environment. And the most important thing about the mucosal immune system or response is that it triggers both innate as well as the adaptive immune response. And since we are dealing with edible vaccines which are consumed through buccal cavity and they enter into your gastrointestinal tract, so I'll be mainly focusing on the mucosal immune system of GIT. Now, what are the components of the mucosal immunity in GIT? Now, the main mucosal immune system, whether it is present in GIT or in respiratory tract or urogenital tract, all these mucosal immune system, they share a basic anatomic organization. And in this organization, they have an outer epithelial barrier that serves essential function to prevent microbial invasion. One. Second, below this epithelial barrier is present a layer of connective tissue which contain different types of cells that are important for innate or adaptive immune response, and these cells include dendritic cells, macrophages, or lymphocytes, that is both B and T lymphocytes. And the third part is the distant draining lymph nodes, which also form an important part of the immune system, and here the adaptive immune responses to the invading microbes are initiated and amplified. Now, as you can see in the picture, the small intestines, which are the major portion for the mucosal immune system in gastrointestinal tract, these small intestines are lined by a continuous epithelial layer, and this epithelial layer, as we discussed, serve as a barrier to the external environment or to the pathogenic microbes or other microorganisms. But on the same time, various kinds of nutrients or the food material, it get absorbed through this epithelial layer into the systemic circulation, and this is how we get the nutrients from the food. Now, as we discussed, below this epithelial layer is a layer of the loose connective tissue, and in case of gastrointestinal tract, this layer of connective tissue is called as lamina propria. And this lamina propria, as I told you that it's a layer of connective tissue, it contains blood vessels, lymphatic vessels, scattered lymphocytes, dendritic cells, macrophages, mast cells that mediate innate immune response, also contains the lymphocytes, that is killer T cells or plasma B cells, which initiates the effector part of the adaptive immune response. Now, all these cells, they are collectively called as Mucosal-associated lymphoid tissue or the MALT tissue. Now, since we are talking about gastrointestinal tract, this MALT or the mucosal-associated lymphoid tissues, when these are present in gastrointestinal tract, they are known as GALT, that is gut-associated lymphoid tissue. So we can say that GALT is a type of MALT. Now, in addition to these lymphoid follicles or the GALT, certain lymphoid follicles are also present below the epithelial layer, and the group of these follicles, lymphoid follicles, is known as Peyer's patches, and these Peyer patches are also considered as a part of GALT. Now, the epithelial lining above these Peyer patches is known as follicular associated epithelium. Since Peyer patches are nothing but these are the lymphoid follicles, and therefore the epithelial cells that are present above these follicles are known as follicular associated epithelial cells. Now, the epithelial lining of the gut, it has different types of cells in it. One is the epithelial cell, second is the goblet cells, which are responsible for the secretion of mucus. Then different types of cells which are known as Paneth cells are also present at the base of the crypts, and these Paneth cells are responsible for the secretion of antimicrobial peptides. So as we discussed that the first line of defense includes the barrier of the epithelial cells as well as certain antimicrobial peptides. So these antimicrobial peptides in GIT are secreted by Paneth cells. Now, these follicular associated epithelial cells, they also contain certain specialized cells which are known as microfold or M cells. Now, these microfold or M cells, they entrap the pathogens or the pathogenic bacteria or antigen, and they pass this pathogenic bacteria or pathogens onto the dendritic cells, and these dendritic cells are lying in close proximity to these M cells. And once these are transferred to the dendritic cells, these dendritic cells take these pathogens, engulfed pathogens, to the Peyer's patches, where the adaptive immune response gets initiated. Or these dendritic cells, they can take these pathogens directly to the mesenteric lymph nodes or the lymph nodes that are present in the GIT. Now, as you can see in this picture, now these epithelial cells in the intestinal villi, they are very tightly packed with the help of transmembrane proteins which are known as occludins or claudins. And these proteins help in forming a tight barrier which acts as the first line of defense. Now, these proteins also form crosslinks with the cytoskeleton of the cells, thereby preventing the entry of the microbes. Mind you that these transmembrane proteins, along with the cross-linkings, they form the tight barrier which prevents the entry of the microbes from the lumen into the systemic circulation. Now this barrier is also supported by a deposition of glycocalyx. Now this glycocalyx is nothing but it's a mixture of glycoprotein and polysaccharides. Now on the outer side of this barrier is present the mucus, which is secreted by the goblet cells, as I've already discussed. Now this mucus is nothing but it is made up of oligosaccharides and glycoproteins, which are known as mucins. And these mucins have an special ability to bind the proteins that are present on the bacterial surface, thereby trapping them and preventing their entry into the cells. And this is how this first line of defense works. Now, in addition to this, as I told you that Paneth cells are there which secrete antimicrobial peptides, and these antimicrobial peptides are of two types, alpha and beta defensins. Now, alpha defensins are further of two types, alpha defensin 5 and alpha defensin 6, and beta defensin are again of two types, BD1 and BD2. And as I told you, both these are secreted by the Paneth cells. Now, these antimicrobial peptides, they kill the pathogenic microbes outside the epithelial barrier itself. But as the microbial load increases, their secretion also increases. Now, apart from these alpha and beta defenses, there is another type of antimicrobial peptide which is known as terfoil factors. And these terfoil factors can be TTF1, TTF2 or TTF3. Now, the first two terfoil factors, that is TTF1 and F2, they prevent the entry of the microbes, whereas the third factor, that is TTF3, it helps in repairing the damaged cell or the cells that are damaged by infection or by any kind of pathogen or any kind of stress. So we can say that the innate immune response of the gastric mucosal immunity is exhibited by the physical barrier, which we have already discussed, that is the tight junctions that are formed by the proteins which are known as occludins or claudins. And the second component of innate immune response in GIT is the chemical barrier, which is formed by the mucus and the antimicrobial peptides, that is alpha and beta defensins and terfoil factors. Third component of the innate response in GIT is the action of toll-like receptors and node-like receptors, that is TLRs and NLRs. Now these TLRs and NLRs activate dendritic cells and macrophages, which cause phagocytosis, and also activate inflammatory response. Now TLRs on binding with PAMPs cause phosphorylation and reorganization of occludins. Now they not only causes the phagocytosis or initiate the inflammatory response, but on binding with PAMPs they also cause phosphorylation and reorganization of occludins, and this reorganization further tighten up the epithelial barrier, thereby preventing the entry of the microbes. So this is basically how the innate immune response works in the gastrointestinal tract. Now we move on to the adaptive response of GIT, or the components of the adaptive response of GIT. Now the adaptive response in gut are initiated by lymphocytes, that is T and B lymphocytes, and antigen presenting cells. And these antigen presenting cells can be dendritic cells or M cells, which present antigen to dendritic cells, and these dendritic cells can further present the antigen to the lymphoid tissue. And these both lymphocytes and APC, that is antigen presenting cells, are closely associated with the mucosal epithelial lining of the bowel and in the mesenteric lymph nodes. Now the nave lymphocytes are those which have not been exposed to any kind of antigen, and these nave lymphocytes are both B and T lymphocytes. These are exposed to antigens in these sites and differentiate into effector cells. So as we discussed that both these lymphocytes, they differentiate and proliferate in adaptive immune response. Another important component of adaptive immune response in GIT is the Peyer's patches. And Peyer patches are nothing but these are the lymphoid follicles that are present in aggregates. And these Peyer patches, if you see in the picture, these Peyer patches have two major parts. First is the subepithelial dome, which is known as SED, and this is nothing but the space between follicular associated epithelial and the Peyer patches, because Peyer patches are nothing but lymphoid follicles, and since the epithelial lining is present above these follicles, therefore it is known as follicular associated epithelial. So the space between the epithelial and these follicles is known as subepithelial dome, and this SED region is filled with T cells, B cells, or the mature B cells which can secrete antibody, and these mature B cells we know are also known as plasma cells, macrophages and dendritic cells. The second portion is the follicle region, and follicle region is further differentiated into B cell zone, which is also known as germinal center, as you can see in the picture, mental zone or the MZ zone, and the T cell zone. Now the germinal center, which is the B cell zone, is further divided into a light zone, which is having resting B cells, which are also known as centerocytes, and the dark zone, which contains the dividing or proliferating B cells, which are also known as centeroblast. Now this germinal center is surrounded by mental zone, as you can see in the picture, and this mental zone is also composed of macrophages, T cells, mature B cells, which are also known as I told you, plasma cells, and they secrete antibodies, and dendritic cells. Now apart from this follicle region also has a T cell zone where T cells are present, and in this zone the population of T helper cell is far more than the T killer cells. So friends, this was about the components of the immune or the gastrointestinal immune system, mucosal immune system, that what are the different components, how the innate mucosal response takes place, and what are the components of adaptive mucosal GIT immune system are there.